Hello, this is a video to walk you through to collect your data from cBioPortal. If you're choosing to follow template research question number 1, which is which genes are involved in patient survivability in insert cancer of your choice. So for this example, I'm going to look at genes involved in glioma, but you can customize this for whatever your research needs are. So starting off on the cBioPortal landing page, the first thing you're going to do is select your data set. So you can do that by coming over here to the side and you can select cancers of whatever interests you. So I said in this example, I'm going to do glioma, so I'm going to click on this CNS/brain. And when I do that, it takes me to all of the patient data sets that are related to the central nervous system and the brain. So I'm going to look at glioblastoma. I still have multiple choices. For this example, I'm going to look at this data set, which comes from the 2020 Pan Cancer Atlas. If you're not sure where to start, I recommend looking at this 2020 Pan Cancer Atlas study, just because it's a very large study. And because it is so large, it's more likely you'll have enough samples to do statistical analyses. You're not required to do this, but it's a good place to start if you're not sure where to start. And if you go through all of this and your data set aren't big enough to do the type of analyses you would like to, you can go back to any of these others and handily, cBioPortal will give you the PubMed link for all of these data sets so that you can properly cite them. All right, so I've selected my patient data set and I'm going to go down here to explore selected studies. So when I click on that, it takes me to a data dashboard related to this patient set, and it's large and it can be a little overwhelming. But for this template research question, since we're looking at genes related to patient survivability, we're going to go down over here to mutated genes. And for this example, I'm just going to click on the 2 genes that are most commonly mutated in this data set. So I select those by hovering my mouse sort of near the name, and that brings up this little magnifying glass icon. So I click that. So I've done that for P10 and for my other most commonly mutated gene, P53. So when I have my magnifying glass icon selected there, it's also going to auto-populate these gene names up in the query box. So with those gene names there, the next thing I'm going to do is click Query. So this will take you to a new window, and cBioPortal has done all of this magic for you. So by default, you're going to start off where it says Oncoprint, and this is one of the pieces of data that you're going to be collecting. So you can immediately go here to download, use that drop-down menu, and download your data, this particular graph in whichever format you choose. This is optional, but I am going to remove some of these tracks because I particularly am only interested in these bottom 2 lines. So clicking on these 3 dots, I can remove the tracks. Let me do that. Alternatively, if you go to tracks over here, you can add and remove tracks based on your needs. All right, so this is the Oncoprint and the way that I like it. And to walk you through these data, we have these numbers here, which are going to tell you how frequently these genes are mutated in this particular patient data set. We also have these color codes, which tell us what types of mutation. And as P10 and P53 are both tumor suppressors, unsurprisingly, we see a lot of deep deletions, we see a lot of truncation mutations, which is what you would expect with a tumor suppressor. So if I was happy with this, again, I would click download and download my file. So the 2 pieces of data you're collecting for this are the Oncoprint, so you have that. And then next is going to be your survival graphs. So to do that, I'm going to go over to comparison/survival. So when I do that, it takes me to this pictorial representation of my data. And by default, cBioPortal has grouped my data into altered. And in this example, it means that these are going to be patients that either their samples have a mutation in P53, P10, or both. And if they're in the unaltered group, there's no mutations in either genes. So I could go forth and analyze my data like this, but I also want to look at the two genes independently. So to do that, I'm going to have to reorganize my data. So I am going to unclick altered and unaltered, and I'm going to click the names of my two genes, so P10 and P53. And patients, I'm not as worried about because there are multiple samples taken from a single patient, but I do want to make sure that my samples don't overlap. So I notice here that I have 61 samples that have a mutation in both P10 and P53. So I'm going to click on that. And if I say create group, I can name that TP53 and P10. So I've created a new group there. I also want to be able to look at just P10 mutations and just TP53. So I'm going to click on these. Again, create a new group, and I'm going to call this TP53 only. I'm also going to do the same thing for P10, so this 126, 10 only. All right, so the new groups that I've created and named are going to show up here at the top. So I will click away this. I want to look at my unaltered group, no mutations, the group that has both mutations, only P53 and only P10. And I know that I've set everything up correctly because if I look at my samples, there is no overlap. So you can see what overlap would look like over in the patient graph. So my data are organized. Now to look at my survival graphs, I'll click over here where it says survival, and this brings that up. So, depending on your sample size, cBioPortal will do the statistical analyses for you. So, in my particular group, I have a p-value of 0.02, so nothing is statistically significant. If I were looking at a data set where things were statistically significant, I would also see hazard ratios. So, you may or may not see that when you pull your data just based on how you have parsed apart your patient samples. If you are unhappy with the way that your survival graph looks, you can always go back a couple of steps and either pull from a different patient data set, so something other than the Pan Cancer Atlas, or you could separate your patient groups a little differently. Perfectly up to you, whatever you choose to do. But if I want to download this, there is a download icon here where I can download these graphs in various file formats, including the raw data. One more trick to show you is by default, you are going to be shown the overall survival. For some data sets, if available, if applicable, you also can click and look at disease specific and progression free. And you can download those the exact same way. All right, so that gets you your Oncoprint and your survival graphs, and you are ready to move forward with template research question 1. So I hope this was helpful and have fun playing with the data.